Hello, hi, I'm Sandhya. I'm your botany lecturer. Today, now we are going to study about cell division. Cell division means division of the cell. So this cell division, cell division. Before going into cell division, let us discuss about the cell cycle. What is the cell cycle? See, all living organisms are made up of cells. Some living organisms are unicellular, they are made up of single cell. Some living organisms are made up of many number of cells, they are multicellular. So for their growth and reproduction, from one cell, they are going to form into two daughter cells. Then these two daughter cells, they are going to grow and then they are going to divide matter. So there is a sequence of events which are taking place from growth to division. So this particular sequence of events by which the cell is going to duplicate its genome, then grow in its size, synthesis of other constituents of the cell, and dividing into two daughter cells. That particular sequence of events is called a cell cycle. So this is a sequential events of the cell where duplication of genome and synthesis of other cell constituents and division of this particular cell into two daughter cells is nothing but the cell cycle. It's a sequence of events which are going to take place for the duplication of the genome, means doubling of the chromosomes, duplication of the genome, then synthesis of other cell constituents and dividing into two daughter cells. So this is about the cell cycle. Then in this cell cycle, what happens? The DNA synthesis is going to take place. The DNA synthesis is going to take place at one specific stage. At one specific stage, the DNA synthesis is going to take place. And whatever replicated chromosomes are there, they are distributed to the daughter nucleus. And all these events are genetic control. This is about the cell cycle. Okay, cell cycle. So this cell cycle can be represented in the form of a pie chart. Okay, this cell cycle can be represented in the form of a pie chart. Take for example, this is a pie chart. Okay, the first stage is G1 stage, then S stage, then G2 stage. This is the, wait a minute. Yes, this is a G2 stage. Okay, and this is a G0 stage. This is the M phase. This whole phase is a M phase. Little bit M. First, this is G1 phase, S phase, G2 phase. This is G0 phase. So first the cell will be 1. Then it undergoes this whole phase. Then it undergoes a division phase. By the end of this, what happens? There will be two daughter cells. Okay, so this whole phase is nothing but interphase. This is the M phase. Okay, so the sequential events which are occurring in a cell from the duplication of the genome and synthesis of other cellular constituents to the division of the cell into two daughter cells. All those events are nothing but the cell cycle. It can be represented in the form of a pie chart. Okay, so the cell cycle can be divided into two. First is interphase. The second one is called as M phase. Okay, so this interphase and M phase. So interphase is somewhat like a resting stage. Resting phase or resting stage. This is the division phase. Division phase. Cell division takes place. Okay, so all these in a human cell, all these all sequential events or the cell cycle takes place within 24 hours. Within one day, the cell cycle is going to complete it. But in yeast, it is about 90 minutes. The cell cycle is 90 minutes. The cell cycle is 90 minutes. Then it is divided into interphase and M phase. Then, so human cell is having a cell cycle of 24 hours. So in this 24 hours, the more duration of period where the cell is going to be in the cell is going to be there for longer duration. The cell is going to be there for longer duration in interphase only. 95% of the cell cycle duration is interphase. The remaining 1 hour it takes place in the remaining 1 hour is taken by the cell to divide in matter. The remaining 1 hour is taken by the cell to divide. So the interphase represents the phase between the two successive mitosis phase or M phase. The interphase represents the two successive M phase. Then 24 hours average duration of cell cycle of a human cell. Then and this particular division lasts only for 1 hour, but 95% of its cell cycle is nothing but the cell will be in the interphase stage. Then this M phase, this M phase is the division phase. It can be mitosis, it can be meiosis. Later on we will study, but first of all we will study about the interphase. Interphase is a phase which is present between two M phase. Means here again M phase takes place, division phase. It will be interphase, then again M phase. So it is a phase in between interphase. It is a phase in between two successive division phase. It is called as a resting phase. Resting phase literally doesn't mean resting. It doesn't mean that the cell is taking rest. Here the cell is preparing itself for the division purpose. The actual cell growth takes place. And the cell is preparing itself for the to undergo division. So here what is happening? The cell growth is happening, the DNA replication is happening, and also other cellular constituents are also prepared in this interphase. This interphase is again divided into G1 phase, S phase and G2 phase. G1 is nothing but the gap phase 1. S is nothing but the synthesis phase. G2 phase is nothing but the gap 2 phase. The G1 phase is in between the M phase and the synthesis phase. Here it is the cell is going to grow. The cell is going to show the growth and matter. Then coming on to S phase. S phase is the synthesis phase. Here in the S phase, this S phase is in between G1 and G2. In this S phase, the DNA replication takes place. The duplication of the DNA is going to take place. That means the genetic content is going to become from 2C to 4C. It is going to become from 2C to 4C. But there is no increase in the chromosome numbers. Only the replication of the DNA takes place. That is, if the initial amount of DNA is denoted as 2C, then at the end of the S phase, that it will become into 4C. It increases to 4C. That means there is no increase in the chromosome number. That means if the cell is diploid in G1 phase, then in S phase, after the completion of S phase also, it will be in diploid. No increase in chromosome, but only the DNA replication takes place. That clarity should be there. Then what happens? Then in G2 phase, it's a gap 2 phase. Here it is in between the synthesis and the M phase. And in this phase, the other constituents which are required for the cell for its growth and for its division, other constituents like proteins are synthesized, and the cell growth is going to be continued. This is about the gap 2 phase. Sometimes what happens, sometimes what happens is some of the cells after dividing, some cells in the adult animals do not appear to exhibit division. So what happens? They'll directly enter into the G0 phase. They'll not show division after maturation. What the cells after growing, after maturation, they'll not show any division. They'll show only maturation. They'll not show any division. They'll enter into a stage called as G0 phase. There will be no G1 phase. They are going to exit G1 phase. What is happening? They do not show any division. What's a very good example is heart cells. That G0 phase is nothing but the quiescent stage. Quiescent stage is G0 stage, where the cells are not going to show any division. Okay, quiescent stage. Okay, it is these cells that do not divide further exit G1 phase to enter an inactive stage called as quiescent stage G0 of the cell cycle. Okay, so these cells that they no longer proliferate unless and unto so dependent on the requirement of the organism. Majorly they will not show any division, but suppose if the organism demands the cells to divide, it will divide. Means the normal heart cells are not going to divide, but if it is in a cancerous cell, if the organism is saying to undergo that one, means if it is a cancerous cell, it may divide and matter. But majorly these are the cells which are going to exit G1 phase, and they enter into an active stage where the cells are not going to proliferate. They will not show any division. Such type of stage is called as quiescent stage or G0 stage or G0 phase. Coming on to now M phase, the divisional phase. Okay, the divisional phase. If this is a somatic cell or a vegetative cell or normal growth, normal vegetative and somatic cells, mitosis is going to take place. Mitosis is a type of cell division. Mitosis is a type of cell division which is going to take place in vegetative cells and somatic cells, where the genetic constitution of the parent cells and the daughter cells is same. Okay, so this is similar, same. Okay, so this mitosis. This mitosis is going to take place both in diploid and haploid cells. This mitosis division is going to take place both in diploid and haploid cells. If the cells are diploid, the daughter cells are also going to be diploid. If the cells are haploid, if the parental cells are haploid, the daughter cells are going to be haploid. So in both the cases, the mitosis is going to take place. Okay. Then they are going to maintain the genetic constitution. Whatever genetic constitution is in the parental cell, the same constitution will be present in the daughter cells also. And from one cell, two daughter cells are formed. And it is also called as equational division. The mitosis is also called as equational division because equally the chromosomes or the genetic contents is divided between the two cells, between the two daughter cells. Equational division. Then this mitosis. This mitosis is again divided into 4 stages. First stage is prophase. First stage. Okay. So what is going to happen in this first stage? First is prophase. Second is metaphase. Third is anaphase. Fourth is telophase. Okay, so first prophase. So what is going to happen here in the prophase? In the prophase, the compaction or initiation or condensation of the chromosomes is going to take place. Okay, here the compaction and condensation of chromatin material, condensation of chromosome material is going to takes place. Chromosome material. In the interphase stage, the genetic material is going to be seen is seen in the form of chromatin reticulum, like this in the interphase. But here in the prophase, this particular chromatin reticulum is going to become compact. It is going to condense into chromosomes. Condensation of the chromosomes is going to be taking place. Then slowly, suppose this is a cell. Okay, slowly the nuclear envelope is going to disappear. Then nucleolus is going to disappear. By the end of prophase, what happens? This particular nuclear envelope disappears, the nucleolus disappears, and the chromosomes are going to be present. The chromosomes are going to be present. They will be floating in the cytoplasm like this. The chromosomes are present. So what is happening here? The chromosome material condenses and organizes to form a motile chromosomes. Then here what happens? If it is an animal cell, the centrioles are going to divide into two, and they are going to synthesize the spindle fibers, mitotic spindle. But if it is a plant cell, the cytoplasm is going to synthesize in the plant cell. The cytoplasm is going to have microtubules that is going to synthesize the spindle fibers. The formation of spindle fibers occurs in the prophase. So first what happens? The nuclear envelope dissociates and the nucleolus dissociates. If it is an animal cell, the centriole divides into two. Duplication of centriole takes place. Then condensation of the chromatin reticulum takes place to form into motile chromosomes. So these are the stages which are going to occur. These are the changes which are going to occur in the prophase. This is the first stage of the divisional phase. And also in this, the double membrane cell organelles like endoplasmic reticulum, Golgi complex, then endoplasmic reticulum, Golgi complex are going to disappear. Endoplasmic reticulum, ER, Golgi complex disappears. That is in the prophase. Like this, the chromosomes are there. Metaphase. This is the second stage. Meta means middle. So what happens here? The initiation of formation of spindle fibers takes place now. Here what happens? In the metaphase, those spindle fibers are going to attach to that particular chromosomes. Those spindle fibers are going to attach to that particular chromosomes. Okay? Chromosomes are there, they are going to get attached like this. Like this. Irregularly. They are arranged. Okay? So in the metaphase, the chromosomes are like this. And the spindle fibers are going to attach to this particular things. Like this. Like this. Okay? So and by the end of the metaphase, what happens by the end of the metaphase? By the end of the metaphase, these particular chromosomes are going to align to that of the equatorial plate of the cell. Okay, and like this, and the spindle fibers are going to attach to the kinetochores of the centromere. Like this. The spindle fibers are going to attach to the kinetochore of the centromeres like this. Okay, so this is about the metaphase. Metaphase means the chromosomes are going to align on the equatorial plate and the spindle fibers are going to attach to the centromere, mainly to the kinetochore where the centromere is embedded. Okay, and here the morphology of the chromosomes can be clearly seen, can be clearly studied also. The metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid, each chromosome connected by its kinetochore to spindle fibers from one pole, and its sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. Okay, so here this is centromere, kinetochore. So one of the spindle fiber from one pole is going to attach to this, and the kinetochore of this side is attached to the spindle fiber of the opposite pole. Like this, this is going to be formed. So what key features are going to occur in metaphase? Spindle fibers are going to attach the kinetochore of the chromosomes. Chromosomes are moved to spindle equator and get aligned along the metaphasic plate through spindle fibers to both the poles. Like this. This is the stage in metaphase. Coming on to anaphase now. What happens in anaphase? The splitting of the centromere takes place. The splitting of the centromere takes place, and they move to the opposite poles. Like this. They move to the opposite poles. Okay. So centromeres are going to split up and chromatids separate. Here the centromeres. So in the anaphase, what happens? The centromeres are going to split up, and they move to the opposite poles. And the chromatids are going to move to the opposite poles. This is about anaphase. Coming on to telophase. In telophase, what happens? In telophase, what happens? The decondensation of the chromatids, the decondensation of the chromosomes is going to take place, and they are going to form into chromatin reticulum. Okay, like this. Decondensation of the chromosomes is going to take place, and they are forming into chromatin reticulum. So here what is happening with the chromosomes cluster at opposite spindle fibers, and their identity is lost as discrete elements. Here discrete elements are present in anaphase, no? Here in telophase, as discrete elements, it is lost, and they get condensed. And form into a cluster-like thing. Then here what happens? The nuclear envelope develops. This is the nuclear envelope develops. The nucleolus develops. Nucleolus develops, and the Golgi complex and ER is going to be developed. So here the division of the nuclei has occurred. Okay. So here what is happening? The chromosome cluster has formed at opposite poles. The nuclear envelope has developed. The nucleolus has developed. The Golgi complex has developed in telophase. So in this telophase is going to be followed by the followed by the cytokines. So this is nothing but the division of nucleus. So division of nucleus is nothing but karyokinesis. The division of the nucleus is called as karyokinesis. The segregation of duplicated chromosomes into daughter nuclei is called as karyokinesis. Now sometimes what happens, most of the times the karyokinesis is going to followed by the cytokinesis. Cytokinesis is nothing but the division of the cell. What is going to happen in this? Like this, the cell is there, no? Here a small constriction is going to be formed. Then this is the nuclei. A cell plate is going to be formed, and this cell plate, which is nothing but the middle lamellum, is going to form. It is going to enlarge and divide, and this particular cell is going to divide into two. So first the cell plate formation is going to take place, that represents the middle lamellum between the two adjacent cells. Then again it divides into two. Sometimes in some of the cells, the karyokinesis is not followed by the cytokinesis. In that case, what happens? In one particular cell, many nuclei will be present. Like for example, in syncytium in a liquid endosperm of coconut. Liquid endosperm of coconut means binucleated condition. Multinucleated condition is seen because karyokinesis is not followed by cytokinesis. That particular condition is called syncytium. It can be seen in liquid endosperm. Free nuclear division takes place, where the cell division doesn't take place. cytokinesis doesn't take place. This is about the mitosis phase, and this division phase is about 1 hour, whereas the whole cell cycle is 24 hours. So 95% of the cell will be in interphase stage only. But when it wants to divide it, within 1 hour it is going to complete its division. This mitosis is also called as equational division, which is going to express in vegetative cells, somatic cells, diploid cells or haploid cells. When the diploid cells are undergoing mitosis, the diploid daughter cells are formed. 2 diploid daughter cells are formed. When the haploid cells are undergoing mitosis, the same haploid cells are formed in the daughter cells. 2 daughter haploid cells are formed. Means same genetic constitution is maintained. That's why the mitosis is also called as equational division. It's also called as equational division. It is divided into 4 stages: prophase, metaphase, anaphase, and telophase. Prophase, first phase. First phase law. What is happening here? The disappearance of the nuclear envelope, nucleolus, Golgi complex, and ER is going to get disappeared. And main thing, what is happening? The compaction of the chromatin reticulum takes place to chromosomes. Then metaphase. Metaphase, what happens? Here the spindle fibers are going to attach to that particular chromosomes. Where they are attached? To the kinetochore of the centromere. Then they'll get aligned onto the metaphasic plate or the equatorial plate. That is metaphase. Those are changes which are going to occur in the metaphase. Anaphase, what happens? The splitting of the chromosome takes place, and splitting of the chromosome centromere takes place, and these spindle fibers will be moving the sister chromatids to the opposite poles, like this. Then in telophase, what happens? In telophase, telophase, this is this, anaphase is this. So telophase, what happens? What is happening here? The chromosome cluster is formed here at opposite poles. Then the nuclear envelope develops, the nucleolus develops, and the Golgi complex ER also develops nicely. Then this is nothing but the division of the cell karyokinesis. This will be followed in most of the cells, will be followed by cytokinesis. In sometimes it will not be followed. The cytokinesis will not be followed by karyokinesis. In such conditions, syncytium condition is going to develop, and that is syncytium condition. Syncytium condition can be seen in liquid endosperm of coconut. Coming on to significance, what for this mitosis takes place? Why mitosis has to be takes place? Yes, mitosis is very important. It has to be taken place in the cells. It is also called as equational division. Why? Because in diploid cells is going to occur, in haploid cells is going to occur, and from one cell 2 cells are occurring. So if the parent cells are diploid, the daughter cells are diploid. If the parent cells are haploid, the daughter cells are haploid. So same genetic constitution is maintained, and it's also useful for the cell repair. Suppose any damage occurs to the cell, so that particular cell is going to undergo division, and it's going to get repaired, and the new cells are going to form. Then new cells are going to form. For cell repair, they are important. Then and also in multicellular organism, for growth purpose, this mitosis plays an important role, and this mitosis is also important for nucleocytoplasmic ratio. It is also important for nucleocytoplasmic ratio. So it is an equational division. If we move on, if we say about significance of mitosis, it is an equational division. If the parent cells are diploid, these are also diploid. These are parent. These are offsprings. If the parent cell is haploid, the daughter cells are also haploid. So same genetic constitution is maintained. That's why equational division. They are going to maintain the nucleus and cytoplasmic ratio. Means the contents of the nucleus and cytoplasm, that ratio is being maintained. It is useful for cell repair. Then in multicellular organisms, for growth purpose, cell growth purpose, it is very much important. Then it plays an important role in the epidermis for the formation of multi-layered epidermis, the lining wall of gut, then upper layer of the epidermis of the gut, blood cells, they'll get replaced and new cells are formed with the help of mitosis. This is about the mitosis. Thank you.